Hello students, welcome to EPG Pathshala. I am Dr. Kapendra from Kirodemal College, University of Delhi. Today we will discuss about the module receptors in neurotransmission. After studying this module, you shall be able to understand how nerve impulse travel from one axon to another axon. Different types of receptors involved in neurotransmission. Learn basic structure of ionotropic and metabotropic receptors. Know mechanism of action of different neurotransmitter receptors at different stimuli. Understand the advantage of signal cascade mechanism. Communication between adjacent neurons occur at specialized regions of the nerve cell termed the synapse. It is mediated by the movement of chemical mediators or neurotransmitters across a small gap that exists between the nerve cells. The neurotransmitter receptor proteins are integral component in the communication between two adjacent neurons protruding into the synaptic cleft and cell cytoplasm. When a neuron is activated, it releases neurotransmitter from synaptic vesicles localized in the presynaptic terminal that diffuses across the gap between the neurons, the synaptic cleft to the postsynaptic membrane. There are different types of neurotransmitters. They are mostly small hydrophilic molecules such as biogenic amines like dopamine, noradrenaline and serotonin, amino acids like GABA, glycine and glutamate, and low molecular weight polypeptides and enkephalin, neurotensin, etc. Neurotransmitters are unable to cross through the hydrophobic neuronal postsynaptic membrane. Instead, they show their effect by interacting with receptors present in the postsynaptic membrane. Each type of receptor has one or more neurotransmitter binding site at which neurotransmitter binds. When a neurotransmitter binds to the correct receptor, it causes opening of an ion channel and form a postsynaptic potential, either excitatory postsynaptic potential or inhibitory postsynaptic potential in the postsynaptic membrane of a cell. Neurotransmitter receptors can be categorized as ionotropic receptors or metabotropic receptors. This division is based on the fact that whether the neurotransmitter binding site and the ion channel are part of the same protein or are belong to different proteins. Ionotropic receptors are the kind of neurotransmitter receptors that contain a neurotransmitter binding site and an ion channel which are components of the same protein. An ionotropic receptor is also considered as a type of ligand-gated channel. In the absence of a ligand, ion channel component of the ionotropic receptor remains closed. When neurotransmitter bind to the ionotropic receptor, it causes the ion channel to open and result in EPSP or IPSP in postsynaptic neuron. Many excitatory neurotransmitter bind to these ionotropic receptors which open cation channels, allow movement of sodium, potassium and calcium ion channels through the postsynaptic cell membrane. However, inflow of sodium is more than calcium ion inflow of potassium outflow. As a result, inside of the postsynaptic neuron became depolarized. Many inhibitory neurotransmitter when bind to ionotropic receptors having chloride channel results in the opening these chloride channels and allow inward diffusion of large number of chloride ions. The inward movement of these chloride ions make inside of the postsynaptic cell to become hyperpolarized. Metabotropic receptors are the type of receptors that contain a neurotransmitter binding site, but it does not have an ion channel as its integral part. However, these receptors are coupled to a separate ion channels through a membrane protein called G protein. When a neurotransmitter binds to these receptors, G protein either directly opens the ion channel or indirectly by activating other molecules such as second messengers in the cytosol, which in turn opens or close the ion channel. Some inhibitory neurotransmitter bind to the metabotropic receptors that are linked to potassium channels and causes opening of these channels. The opening of these potassium channels increases outward diffusion of large number of potassium ions, which make inside of the post-synaptic cell to become hyperpolarized or more negative. The same neurotransmitter can act as excitatory at some synapses and inhibitory at other synapses, depending on the structure of receptor at which it binds. For instance, at some excitatory synapses, acetylcholine binds to the ionotropic receptor, which subsequently generate EPSPs in the postsynaptic cell, whereas at some inhibitory synapses, acetylcholine binds to metabotropic receptors and subsequently generate IPSPs in the postsynaptic cells. Ligand-gated ion channels or transmitter-gated channels. These are ionotropic receptors are known as transmitter-gated ion channels or ligand-gated ion channels are membrane spanning proteins having 4 or 5 subunits that close together to form a pore. In absence of ligand, the pore remains closed. When neurotransmitter bind to specific site of the channel, it brings a conformational changes in the subunits within few milliseconds and causes the pore to open. Transmitter gated channels generally do not show the same degree of ion selectivity as do voltage gated channels. Acetylcholine gated ion channels at the neuromuscular junction are permeable to sodium and potassium ions when these open channels are permeable to sodium ions, bring the membrane potential toward threshold for generating action potential and depolarize the postsynaptic cells. This effect is said to be excitatory. A depolarization of transient postsynaptic membrane caused by the presynaptic release of the neurotransmitter is known as excitatory postsynaptic potential or EPSP. For example, synaptic activation of acetylcholine gated and glutamate gated ion channels causes EPSPs if the neurotransmitter gated channels are permeable to chloride ions. The net effect will be to hyperpolarize the postsynaptic cell. Chloride equilibrium potential is negative as it brings the membrane potential away from the threshold for generating action potential. This effect is known as inhibitory. The presynaptic release of the neurotransmitter causes transient hyperpolarization of the postsynaptic membrane called inhibitory postsynaptic potential or IPSPs. For example, synaptic activation of glycine-gated or GABA-gated ion channels. Basic structure of ligand-gated ion channels. The most deeply studied ligand-gated ion channels is the nicotinic acetylcholine receptors, which admit both potassium and sodium ions. This receptor is best known for its role in the synapses between motor neuron and skeletal muscle cells. A single channel can be a sensitive detector of chemicals and voltage. It can regulate the flow of large current with great precision, and it can also filter and select very similar ions, and that can be regulated by other receptor systems. The size of each channel in muscle plasma membrane is about 11 nanometer long, through which acetylcholine causes opening of a cation channel in the receptor and can transmit 15,000 to 30,000 sodium or potassium ions in 1 millisecond. The acetylcholine receptor from the skeletal muscle is a pentameric protein subunit arranged like the staves of a barrel to form a single pore through the membrane. It consists of 4 different types of polypeptide designated as alpha, beta, gamma and delta. A pentameric unit is made from 2 alpha subunit and 1 beta, gamma and delta each. The alpha, beta, gamma and delta subunits have considerable homology sequences. About 35% to 40% of their residues in any 2 subunits are similar. Each molecule has a diameter of about 9 nanometer, and out of which about 6 nanometer protrudes into the extracellular space and about 2 nanometer into the cytosol. Each alpha subunit consists of one acetylcholine binding site and simultaneous binding of acetylcholine to both alpha subunits is necessary for opening the channel. The nicotinic acetylcholine receptor on neuron is also a pentamer. However, unlike the muscle receptor, most of them are comprised of only alpha and beta subunits. Most of the other ligand-gated channel in the brain are also thought to be pentameric complexes having close similarities to the nicotinic acetylcholine receptor. The most important exceptions are the glutamate-gated channels, whose structure resembles that of potassium channels, and this led to the hypothesis that both glutamate receptors and potassium channels evolved from a common ancestral ion channels. The most interesting variation among channel structures are the one that account for their differences. Different transmitter binding site make one channel to respond glutamate while another respond to GABA. Certain amino acid around the narrow ion pore allow sodium and potassium ions to flow through some channels, calcium ion through others, and only chloride through yet others. Glutamate gated channels in the mammalian brain. Hippocampus is the region associated with many types of short term memory. Postsynaptic cells of certain hippocampal neurons receive inputs from hundreds of presynaptic cells. In long term potential, a burst of stimulation of the postsynaptic neuron make it more responsive to subsequent stimulation by presynaptic neuron. For example, stimulation of a hippocampal presynaptic nerve with 100 depolarization acting over only 200 milliseconds causes an increased sensitivity of the postsynaptic neuron that lasts few hours to days. Changes in the response of the postsynaptic cell may underlie certain type of memory. 2 types of glutamate gated cation channels in the postsynaptic neuron participate in the long term potentiation. 3 glutamate receptor subtypes are AMPA receptors, that is amino 5 hydroxy 5 methyl 4 isozazole propionate, NDMA receptors, that is N methyl D aspartate, and canite receptors, which mediate much of the synaptic excitation in the central nervous system. The AMPA gated and NMDA gated channels mediate the most of the fast excitatory synaptic transmission in the brain. kainate receptors also present throughout the brain, however, their functions are not clearly understood. AMPA-gated channels are permeable to both sodium and potassium ions and are not permeable to calcium ions. The activation of these channels at normal negative membrane potential allows diffusion of sodium ions into the cell, result in rapid large depolarization. Hence, AMPA receptors at CNS synapses mediate excitatory transmission similar to nicotinic receptors that mediate synaptic excitation at neuromuscular junctions. AMPA receptors present along with NMDA receptors at several synapses in the brain. So, most glutamate mediated EPSPs have components contributed by both receptors. NMDA gated channels causes depolarization of nerve cells by allowing diffusion of sodium ions. However, they differ from AMPA receptors in two ways. NMDA gated channels allow entry of calcium ions and sodium ions. It causes inward ionic current through NMDA gated channels that is voltage dependent. For opening of the glutamate ion channels, glutamate must be bound and membrane must be partially depolarized. In this way, the NMDA receptors function as a coincidence detector, that is, it combines activity of the postsynaptic cell which is reflected in its depolarized plasma membrane as a neurotransmitter is released from the presynaptic cells, generating a cellular response higher than effect caused by glutamate release alone. As postsynaptic cell becomes sensitized, it takes fewer action potential in presynaptic neurons to induce depolarization in the postsynaptic neuron, or the synapse learns to have an enhanced response to the signal from the presynaptic cells. Calcium ions from presynaptic region elicit the release of neurotransmitter, and postsynaptically it activates many enzymes as well as control the opening of different channels. Excessive release of calcium ions can also cause cell death. It occurs due to higher concentration of glutamate which activates at different types of receptors and allow excessive flow of sodium, potassium and calcium ions across the membrane, and neural cells die due to overexcitation, a process called excitotoxicity. The NMDA subtypes of the glutamate gated channel play critical role in excitotoxicity because it allows the entry of calcium ions. Neurons death occurs because of swelling of the cell due to excessive water uptake and stimulation of intracellular enzymes by calcium ions which destroy lipid, proteins and nucleic acids. GABA-gated and glycine-gated channels. In the vertebrate central nervous system, synaptic inhibition is primarily mediated by two amino acids, glycine and gamma aminobutyric acid, that is GABA. The latter is formed by the loss of carboxyl group from the glutamate. The concentration of GABA in the human brain is about 200 to 1000 times higher than other neurotransmitters like norepinephrine, dopamine and acetylcholine. Glycine is considered as major inhibitory neurotransmitter in brainstem and spinal cord, whereas GABA prevails elsewhere in the brain. Both GABA and glycine are responsible for activating the ligand-gated chloride ion channels. Both inhibitory GABA A and glycine receptors have very similar structure as that of excitatory nicotinic acetylcholine receptors. However, GABA A and glycine are selective for anions, whereas nicotine acetylcholine receptor is selective for cations. Each receptor has subunits at which transmitter bind and beta subunits at which no transmitter binds. The opening of chloride channels tend to drive the membrane potential towards the chloride equilibrium potential, which is slightly more negative than the resting membrane potential, and therefore, larger increase in the sodium permeability will be required to depolarize the membrane. The permeability of the GABA or glycine toward chloride ion is induced very rapidly in a fraction of milliseconds. However, it lasts for a second or more. Therefore, glycine or GABA induces a quite long lasting inhibitory post-synaptic responses. Higher duration of synaptic inhibition can causes a loss of consciousness and coma, and too low inhibition leads to a spasm. Hence, it is more important to regulate synaptic inhibition in the brain. Therefore, GABA A receptors has several sites other than its own site where chemicals can severely modulate its function. For example, two classes of drug, benzodiazepines and barbiturates, bind to their own site on the outside face of the GABA A channel. These drugs do not show any significant effect when they are bind to the site in absence of GABA. However, in the presence of GABA, benzodiazepines increases the channel opening frequency and barbiturates increase the duration of channel openings. It results in more inhibitory chloride current and higher IPSPs. These drugs are specific for the GABA A receptors and do not have any effect on glycine receptor function. G protein coupled receptors. There are several subtypes of G protein coupled receptors in the non-neurotransmitter system. The synaptic transmission is mediated by amino acid and amine neurotransmitter by acting on G protein coupled receptors. The transmitter action involves following three steps. First, neurotransmitter bind to receptor which is embedded in the post-synaptic membrane. Second, the receptor activates G proteins which freely move along the intracellular phase of the post-synaptic membrane. Third, these activated G proteins activate effector protein that can be G protein gated ion channels or enzymes which synthesize second messengers. These second messengers activate additional enzymes in the cytosol that regulate ion channel function and can alter cellular metabolism. The G protein coupled receptors are often referred as metabotropic receptors. Structure of G protein receptors. Most G protein coupled receptors are simple and show variation on a common plan. It consists of a single polypeptide having 7 membrane spanning alpha helices. Out of these, 2 extracellular loops of the polypeptide form transmitter binding sites. The structural variation in this loop will determine bind of different neurotransmitters, agonists and antagonists to the receptor. 2 of the intracellular loops can bind to and activate G proteins. Structural variations will determine which G proteins and consequently which effector system will be activated in response to the transmitter binding. Functioning of G proteins. G proteins form common link in most signaling pathway that begins with a neurotransmitter receptor and finish with effector proteins. There are several transmitter receptors as compared to G proteins. Therefore, some G proteins can be activated by several receptors. The functioning of G proteins have same basic mode of operation. First, a G protein consists of 3 subunits alpha, beta and gamma. During resting state, a GDP molecule is bound to the G subunit of G protein and the whole complex floats on the inner surface of the membrane. If GDP bound G proteins strike into the suitable receptor and if that receptor has a transmitter molecule bound to it, then G protein exchange its GDP with GTP, that is pick up from the cytosol. Third, this activated G protein bound with GTP splits into 2 parts, the G alpha subunit having GTP and the G beta gamma complex. These split subunits move on to influence different effector proteins. Fourth, the G alpha subunit also behaves as an enzyme which subsequently breaks down GTP into GDP. Therefore, G alpha ultimately terminates its own activity by breaking down the bound GTP into GDP. Fifth, after that G alpha and G beta gamma complex again come back and join together to start another cycle. Some of the G proteins exhibit the stimulating effect, and it was found that other G proteins could inhibit these same effectors. Thus, the G protein can be subdivided as GS designated the stimulatory G protein and GI designating the inhibitory G protein. Activated G proteins exhibit their effects by binding to either G protein gated ion channels or G protein activated enzymes. As the first route do not involve any other chemical intermediaries, sometimes it is also called as shortcut pathway. A variety of neurotransmitters uses the shortcut pathway from receptors to G proteins to ion channels. One of such example is cardiac muscarinic acetylcholine receptors that activate G proteins. Binding of acetylcholine transmitter to the muscarinic acetylcholine receptor in heart muscle generate a slow inhibitory response. When cholinergic nerve in the heart muscle is stimulated, it causes a long-lived hyperpolarization of the membrane which slows down the rate of heart muscle contraction. The muscarinic acetylcholine receptor is a G protein receptor which after activation lead to opening of potassium ion channels and subsequently causes hyperpolarization of the plasma membrane. The muscarinic acetylcholine receptor consists of 7 transmembrane alpha helices. Binding of transmitter to the receptors activate a trimeric transducing G protein and then the released G beta gamma subunit directly binds to and open potassium channels. The potassium ion channels opened immediately when G beta gamma subunit is added in the absence of acetylcholine. The cardiac muscarinic receptor explains the way in which G protein coupled receptors affect ion channels where the activity G beta gamma subunits bind to the channel protein. Another example is neuronal GABA B receptors coupled by potassium channels. Shortcut pathways are fastest of G protein coupled system, but not quite as fast as a transmitter gated channel, which uses no intermediary between receptor and channel. However, it is faster than the second messenger cascades. A G protein diffuses within the membrane, so it cannot move very far. Therefore, it affects only nearby channels. Second messenger cascade, G proteins also have their effect by directly activating some enzymes that can trigger an elaborate series of biochemical reactions that ends in the activation of other downstream enzymes which can modify neuronal function. There are various second messengers which is present between the first enzyme till the last enzyme. This whole process that couples the neurotransmitter through multiple steps to activate downstream enzymes is known as second messenger cascade. The cyclic AMP, a second messenger cascade, is started with the activation of NE-beta receptor that activates stimulatory G protein. It further proceeds to stimulate the membrane bound enzyme adenylyl cyclase. The adenylyl cyclase converts ATP to cyclic AMP. The subsequent rise of cyclic AMP in the cytosol triggers a specific downstream enzyme called protein kinase A or pKa. Most of the biochemical processes are regulated with a push-pull method in which one stimulates them and other inhibit them. The activation of a second type of NE receptors, that is alpha 2 receptor, leads to the activation of GI or inhibitory G proteins. The GI suppresses the activity of adenylyl cyclase, and this effect can take primacy over the stimulatory system. Some messenger cascades can branch. For example, activation of various G proteins can stimulate phospholipase C or PLC, an enzyme that floats in the membrane like adenylyl cyclase. PLC acts on a membrane phospholipid PIP2 or phosphatidyl inositol 4,5-biphosphate and split it into two molecules named diacyl glycerol or DAG and inositol 1,4,5 triphosphate or IP3, which act as a second messenger. DAG is a lipid soluble molecule that stays within the plane of the membrane and activates a downstream enzyme called protein kinase C or PKC. Whereas other molecule IP3 is water soluble that moves away in the cytosol and bind to the specific receptors on SER or smooth endoplasmic reticulum in the cell. These receptors are IP3 gated calcium channels that cause the organelles to release stored calcium ions. Elevation in the cytosolic calcium ion can trigger widespread and long lasting effects. One effect is activation of the enzyme calcium calmodulin dependent protein kinase or CAMK. This is an enzyme implicated in the molecular mechanism of memory. The advantage of signal cascades. The important advantage of signal cascade is the amplification of several ion channels. For example, activation of one G protein coupled receptor can lead to the activation of several ion channels that occurs at several places in the cascade. A single neurotransmitter molecule bound to one receptor and that activates about 10 to 20 G proteins. Further, each G proteins can activate an adenylyl cyclase which can make several cyclic AMP molecules that can activate many kinases. Each kinase can further phosphorylate many channels. If all components of the cascade were tied together in a clump, signaling would become very limited. The use of small messengers such as cyclic AMP can diffuse very quickly and allows signaling at a distance over cell membrane. Signal cascades can also generate long lasting chemical changes in the cell, which may form the basis for a lifetime of memories. Autoreceptors, in addition to the postsynaptic density, neurotransmitter receptors are also found in the membrane of the presynaptic axon terminals. The presynaptic receptors, which are sensitive to the neurotransmitter released by the presynaptic terminals, are called as autoreceptors. Usually, autoreceptors are G protein coupled receptor that stimulate second messenger formation. The effect of activating these receptors may vary. However, a common effect of autoreceptor is the inhibition of neurotransmitter release, and in some cases it also helps in neurotransmitter synthesis. This allows a presynaptic terminal to regulate itself. Autoreceptors appear to function as a sort of safety valve to reduce release when the concentration of neurotransmitter in the synaptic cleft becomes too high. So, students, let us summarize what we have learned in this module. The receptor proteins are central components between two adjacent neurons during the communication. Due to inability of neurotransmitters to cross the postsynaptic membrane, they respond by interacting with receptors present in the postsynaptic membrane. Neurotransmitter receptors can be categorized as ionotropic receptor or metabotropic receptors. Ionotropic receptors contains a neurotransmitter binding site and an ion channels which are component of the same protein, whereas metabotropic receptors contain a neurotransmitter binding site, but it does not have an ion channels as its integral part. Ligand-gated ion channels are ionotropic receptors which are membrane spanning proteins having 4 or 5 subunits that close together to form a pore. The most studied ligand-gated ion channel is nicotinic acetylcholine receptor, which admit both sodium and potassium ions. It is an important receptor which communicates in the synapses between skeletal muscle and motor neurons. Most of the other ligand-gated channel in the brain are also thought to be pentameric complexes having close similarity to the nicotinic acetylcholine receptor. The most important exceptions are the glutamate gated channel, whose structure resembles that of potassium channels. There are different G protein coupled receptors subtypes which involve binding of neurotransmitter. The G protein coupled receptors are often referred as metabotropic receptors. Various neurotransmitters use the shortcut pathway for communication of information from receptor to G protein, then to ion channels. Cardiac muscarinic acetylcholine receptor display shortcut pathway that activate G protein. G protein activates some enzymes which trigger series of biochemical reactions to activate downstream enzyme called as second messenger cascade. The important advantage of signal cascade is the amplification of several ion channels that can generate long lasting chemical changes, causes the basis for a lifetime of memories. Thank you.